Hello, my name is Xiao Peng, and it is my pleasure to introduce you to GenIA, the Genetic Immunology Advisor Database. Our team created GenIA as a way to help our community keep track of the constantly expanding dimensions of immunogenetics knowledge that is being reported over time. But GenIA really can be a resource for any kind of rare disease because what it does is it endeavors to catalog and collect all of the different kinds of knowledge that might be relevant to a patient or a condition in a uniform, harmonized, and as structured as manner as possible so that people can find all of the different kinds of information that they might need, regardless of whether they're a researcher, a clinician, a patient advocate, a patient themselves, or any other person who has a stakehold in this domain. This is the user interface for GenIA, and you can see the URL in the bottom right-hand corner. Anyone who has an academic position of any kind can apply for a free login. And if you're interested in curating with us, we can also enable you to have access to the curatorial user interface. So what is GenIA? Well, GenIA has a lot of different moving parts to it. Of course, gene-disease relationships are the integral part of the data we store, but we also store gene-specific data, variant-specific data, all of the literature that has been published on these conditions and these genes, functional and clinical and immunophenotypic data. And we try to do this using as many different ontologies as possible through the use of expert manual curation and now some AI machine learning tools as well. And a number of people also use GenIA as a way to have a de-identified patient registry platform if they do not have such a platform in their own institution. When you put all of this together, GenIA can also feed into a tool such as Phenia, which enables people to search for a genetic differential for the phenotypes that their patient presents with. So some of the examples of the kind of information GenIA can provide are shown here. We're taking STAT3 as the gene as an example, and you can see all of the associated gene disease conditions reported for STAT3 that have been cataloged, whether germline or somatic. You can see the variants that have been curated, as well as the diagnostic paradigms and pitfalls people should be aware of when trying to test for STAT3 relevant mutations, and all of the references from which our data has been derived. In addition, you can see the lists of clinical findings of patients where we really truly try to interrogate whether each feature is present, absent, or just unexamined. We enable you to see the pedigrees and relationships between all the patients that are cataloged, as well as the papers in which they may be multiply reported. We also try to report any treatment options and outcomes that have been published in the literature. And you can see here we also have a nice diagram that shows the age of onset of symptoms for the patients in our catalog. We use a lot of different kinds of ontologies so that you can easily navigate between parent and child terms and across different kinds of ontologies. We also have a lot of data on objective counts, cell counts, reference range values, functional and immunophenotypic markers. Our variant database enables you to garner more information about the variant itself and also links out to other kinds of data aggregation and resources on variant information. You can see here as well that any functional data or any patients with that specific variant are also reported. We also catalog any available clinical grade or research grade assays that we know of that have been linked to that gene or that disease so that you can have a way to look for tests that might be relevant to a VUS of interest that you have. So in summary, when you put all of this together, we really use a subject-centered model to store multidimensional, as comprehensive as possible data on immune-driven gene-disease relationships. This enables you not to have to choose between genotype-centric tools and phenotype-centric tools. In addition, this also enables you to store many different kinds of data for your patients if you happen to be interested in using GenIA as your own patient registry or repository. We have a lot of people who've joined the GenIA community, and without their efforts, it would be difficult for us to continue to be able to catalog all of these many gene-disease relationships. So if you are interested in curating your favorite gene or group of genes and diseases of interest, please do write to us, and we can figure out a way to make our collaboration work for you. We also provide increased resources for anyone who may need to download information from the GenIA database. Examples of how others have used GenIA for analysis and research are shown here. So some people might be very interested in what clinical phenotypes distinguish certain conditions from each other. Others may want to know what the proportion of de novo versus inherited mutations are for genetic counseling. Similarly with disease penetrance, or understanding on a more granular level the natural history of patients with specific diseases. We also provide information that enables you to rapidly see how patients have responded to different kinds of therapeutic interventions, whether that is small molecule therapies or any kind of management changes. We also provide, as I said before, very granular patient level data. So you can truly compare what the natural histories and courses that people have experienced across different disease conditions and within a disease cohort look like. And you can even see whether or not diagnostic delays have been improved over time as more patients have been identified for a particular disease. We also enable you to generate rapid heat maps that can show the different ways in which a particular gene and its variants have been functionally tested in vitro or characterized in patient cells, and see what the assays people have used are and what the results of those assays might be. This is Fenya, which I mentioned before. It enables you to quickly develop a genetic differential for your patient's conditions and phenotypes of interest. And again, because GenIA is an international collaborative effort that's not owned by any individual group, we really welcome any potential additional curators who may be interested in working with us, because really this tool is made for the entire community. Thank you so much for listening. Here are some papers associated with our database, and please feel free to reach out if you're interested in using or contributing or have any ideas for how we can improve GenIA for the community. We also want to mention that this is the curator interface, and so if you do end up being interested in becoming a curator, we will make a separate session for you to be introduced to this particular interface. Thank you so much for your time, and hope that you will enjoy the use of GenIA.